I'm going to talk today about oncolytic viral immunotherapy. And what I'd like to do is take you from the very beginning all the way up to the present and even into the future with some things that a number of laboratories are exploring and but are not yet published. So up top are my contact information if anybody wants to contact me. And down here are the various disclosures or conflicts of interest. So, as we mentioned, I'm a neurosurgeon, but back when I was a medical student, I took time off and did a fellowship in virology, and that's how these kind of come together. And it's unusual to start a therapy in brain tumors. Most cancer therapies start in the periphery, and the last thing they do is study tumors in the nervous system because of potential toxicity. But the reason it started in neurosurgery, we had a seemingly unsolvable problem of glioblastoma. And glioblastoma is basically like a bunch of other cancers. It has a very short survival time. And it has cellular characteristics similar to other cancers, intratumoral cellular genomic heterogeneity, cancer stem cells, mitotic activity, diffuse infiltration, necrosis, angiogenesis, and it's in a highly immunosuppressive environment. Our initial explorations were with replication incompetent viruses such as retroviruses, but they were very inefficient. Having the work I had done previously in the fellowship was with Paul Black on SV40, simian virus 40, which is a DNA virus that contaminated the polio vaccine that many Americans and people internationally were given. So there was a big interest in it. And through this, I began to look at how viruses work. And poliovirus is very interesting from an evolutionary standpoint. It goes into your gut, it gets absorbed by the system, it goes throughout the body, but it only kills the anterior horn cells of the spinal cord, paralyzing them. But it doesn't affect the sensory neurons, it doesn't affect cognition. So very specific in terms of how it can kill cells. And we were entering an era of genetics and molecular genetics. And the idea was, could we make a virus that would specifically kill a cancer cell without harming the normal cell? And in our case, we were talking about brain. So the basic concept is shown here. We were making an oncolytic virus that's genetically engineered, so it goes in the tumor cell, makes multiple copies, spreads, kills cells, and as I'll show you later, leaves these antigens. But when it goes into a normal cell, it's genetically engineered in a way that doesn't replicate. And that was the basic concept. Now, when we looked at the variety of viruses, both RNA viruses, DNA viruses, small ones, large ones, and finally settled on herpes simplex virus type 1. Now, we stayed away from the RNA viruses because they're relatively unstable. And we wanted a large virus so we could potentially put various gene inserts into it. And as I say, we settled on HSV1. Now, we had done studies both with replication incompetent retrovirus and even published a study on replication competent retrovirus. But because of the RNA instability, small package size, inefficiency, secondary events due to integration, and lack of antivirals at the time, we thought that in this HIV/AIDS era back in the 1980s and 90s, the FDA was never really approved a replicating retrovirus. So we settled on herpes virus for a number of reasons. DNA is very stable. It's a large size, and you can take out 30 kb and still put other genes in and have it replicate. It doesn't integrate into the genome. We knew that herpes could spread in the brain, and we were interested in brain tumors. And it comes in what I call multiple flavors. You can have a replicative virus, you can have a defective virus, you can even have a latent virus. And so the potential for using this, in this case, I'm going to talk today about cancer, but the potential for using a virus such as this for other neurologic diseases seem reasonable. You can even make herpes viruses that will go antegrade in a neuron, other ones that will go retrograde. So it will be very useful potentially for various neurological diseases. And very importantly, antiviral drugs are available. So if we had a problem, we could turn it off with acyclovir and ganciclovir, et cetera. So this would be useful. So this is the first publication we had, as mentioned by the introduction in Science 91. And basically, The virus it was called VLSPTK. DL meaning deletion, S and P were the two restriction sites, and TK was the thymidine kinase gene. And the basic concept was herpes virus needs thymidine kinase to replicate its DNA. Cancer cells have a lot of thymidine kinase because they're replicating all the time. Brain cells do not because they're post-mitotic for the most part. So the basic idea was to make a virus that would replicate in the brain tumor cells, but not in the brain. And we could show in animals, this is now, and this is important, it's now in mood mice using human tumor. And we could show that survivorship increased as you increased the dose of the virus, okay? And there was no brain toxicity. So we said in this article that virus could be genetically engineered to kill cancer cells without harming normal cells, in the state glioblastoma in the brain, and mentioned various mechanisms of direct viral cell killing, plus new antigens, immunological rejection, et cetera. And I'll go through a lot of that as we go forward. But this was the basic concept. So the VLSPTK was not taken into human trial because, again, the primary antiviral would not work because acyclovir or valcyclovir worked through thymidine kinase, and we took the thymidine kinase gene out. So we wanted to make a virus that was TK positive. And we made this virus called G207. And this was published in Nature Medicine in 95. Now, this is the herpes genome. Basically, G207 is missing both copies of gamma 34.5 and has an inactivation of ICP6. And 34.5 is the major neurovirulence gene. So you take it out and you don't get herpes encephalitis. And ICP6 is a large subunit of ribonucleotide reductase, and deletion selects for replication and dividing cells. So, and this was now TK positive, so it would be sensitive to acyclovir, vanciclovir, valcyclovir, and it had two widely spaced mutations. So even if you had one of them mutate back, you would still have a very inactivated virus. Now, G207 had an initial trial. Jim Marker and a number of other people published in 2000. This was direct injection into the brain tumor, one at one time. And that's important to remember. And there was no evidence of herpes encephalitis by MRI or clinical exam. There were 3 patients where we were concerned about it, and biopsy showed no herpes encephalitis. MRI treatment responses in about a third of the patients. There was one possible histologic complete response and 2 long-term survivors. Chronic toxicity did not occur. It was also clear that oncolytic viral injection is generally not cured. So people have recurrences, people still die from glioblastoma. There was still more to be done, although this was a very exciting first step. The next thing was this concept of in situ vaccination, and this is really where oncolytic viral immunotherapy started. So as I mentioned to you, in those initial studies, we used mood mice and human tumors. They were incompetent. We now began to study what would happen if you had a virus in an immune-competent setting. And in a couple of papers that were published around 1988, 1989, we showed it was possible. So in an immune-competent animal, if you inoculated the tumor on one side, you got a reduction with the virus compared to control. But you also got a reduction in the non-inoculated side. Not as prominent as in the inoculated side, but still very prominent and very statistically significant. And we could show there was no virus in this non-inoculated tumor. So the virus wasn't getting transferred to the other tumor, but we could show the virus had to grow in the tumor that it was inoculated in. If you inoculated in the skin next to the tumor, it didn't work. We could show the tumor antigen involved. So if you took the antigen from this tumor, transferred it to other cells, you could kill them. And we could show that lymphocytes were necessary. So that became the beginning of this so-called oncolytic viral immunotherapy. Now, we next showed that you could improve this by putting various cytokines to stimulate the immune system. And one paper, IL-12, and another paper, GM-CSF. And this has been used with various strategies by a number of investigators at this point. So here was the concept. In this paper with IL-12, we used G207, the virus we were working with at the time, in combination with a defective virus expressing either LAC-C or IL-12. And you can see here the mock, which is no virus, versus the virus alone with IgG versus the IL-12 in both the inoculated tumor and the non-inoculated tumor, showing that the expression of IL-12 in context of oncolytic virus would actually improve therapy. And the same was shown with GM-CSF. This is at various doses, showing that at a higher dose you get a more profound response in the inoculated and in the non-inoculated tumor. And this has gone on. GM-CSF, as you know, is used in TVEC and in a number of other viruses that we'll mention as we go forward. So the next question that we asked was, what about prior exposure? Because more than half the population is HSV positive and have antibodies to HSV. So we took mice and inoculated them in the footpad at the time and raised antibodies to herpesvirus, and then we tested the concept of killing foods. And we conclude that seropositivity did not deleteriously affect the efficacy of intratumoral therapy. And then what we said was, okay, what if in fact having a positive effect did not matter with intratumoral therapy? Could you give multiple injections? So could you now think of this as a vaccine, an in-situ vaccine, where you give one dose and you have booster doses? And what we did was take that concept, it was 6 doses at 10-fold less than the single dose. So here you see no virus, here you see a single dose, and here you see multiple BSK6 doses that are 10 times less. So you're basically getting overall 60% less, 60% of this amount of virus. So you're doing it with booster shots, and you get a much more profound effect. This is tumor volume. You can see literally it's not growing, and you see a much more profound survival effect. So we thought that this multiple inoculation should be considered when we go to clinical evaluation. And Ali Shalabi was a student at the time, but please note that Smoky Toto was a postdoc and was in the middle author in this paper, and that becomes important as we go forward. So since that time, G207, this first virus that we took into clinical trial, has had extended phase 1 studies. 50% of people had radiologic responses. Tumor tissue revealed significant CD4, CD8 immune cells. Patients could be treated sequentially multiple times without toxicity. Resistance did not occur. Patients were retreated with radiographic response after each treatment. And survival was superior to published data in this patient population. And this has now been shown in multiple studies. G207 is currently in use by Freeman and Marker at University of Alabama in pediatric high-grade gliomas. One of the papers that they published in the New England Journal of Medicine had 12 patients, one-time treatment of 10^7 or 10^8 plaque-forming units, plus a very small dose of radiation because they had shown in other studies that radiation sort of stimulated viral growth. And they had an increase in overall survival, particularly in the seronegative patients. Now, this is controversial. Nino Fiocca recently had a paper showing that he had a better response in seropositive patients. I would say the N is low in both of them, so they need to be more extended. And it may have to do with how many doses of virus is given, the type of virus, et cetera. But in all cases, a delayed response was seen, and we've seen this over and over. And there are increases in various lymphocytes at really long-term period. And again, this was just a one-time dose. But let me show you. Here's the delayed MRI response, for example. Here's a child before treatment. You can see the tumor here. And after 1 month, 5 months, 9 months, 12 months. And then you get the cystic degeneration that progresses and the tumor is smaller and the patient's doing well. And there's less edema on T2. And that's been consistently seen in all studies. And there's prolonged intratumoral immune response. So, for example, here's patient 1, 6, 9, 10, and here's the time, 3 months, 9 months, 5 months, 3 months. And you can see these lymphocytic infiltration that extends even 9 months, 5 months later. So you get this profound immune response, and this is from basically from one dose. Now, the next step was development of a virus called G47 Delta. G47 Delta is basically G207 with one additional mutation in a gene called ICP47. Now, why was this done? It was done because we were looking at putting cytokine genes in IL-12, GM-CSF, and we wanted to get an immune response. And AC247 inhibits MHC class 1 presentation in humans. So we thought the deletion would overcome this and improve viral immunotherapy when we cytokines. Unknown to us at the time, the deletion also increases oncolytic activity, which I'll show you, and importantly, in a certain subset of cells. So a few years later, Dirks, who's seen in colleagues in Canada, described these cancer stem-like cells. And normally, cells were grown in monolayer. In this case, these are brain tumor cells that are grown in spheres, neurosphere-like culture. And these are thought to be the cause of recurrences. They're thought to be the sort of the germinal cells within tumors. And basically, they form tumors at 1,000 less cells versus cells growing typical monolayer. So here are two equivalent survival lines. But the difference is the black one is our tumors growing, cells growing in fetal calf serum, and there's this 10 to the 4 cells. The 10 to the 1 cells of these glioblastoma stem-like cells will have the same killing curve. So basically, these were more efficacious cells. And what we found out, and unknown to us at the time G47 delta was made, G47 delta replicates in glioblastoma stem cells, whereas G207 did not replicate very well. So here are two different glioblastoma stem cell cultures, MDG8 and B274. And if you put G207 in, you basically get the same amount or less out. If you put G47 delta in, you get an increased curve and you get more out. So you get a continuing replication. And that's shown here visually in G47 delta expressing GFP. And you can see at 24 hours, the green and then the whole neurosphere is colored. And we didn't know for a long time why this was the case. And as I said, it was a serendipitous finding. But Paul Peters was a student in the lab a number of years later, and he showed that the deletion overcame a block during translation of two late herpes virus genes in the glioblastoma cancer cells. And that was the reason it didn't grow, and G207 didn't grow, but G47 delta did. And we thought that was important. So G47 delta will kill the stem cells, not just glioblastoma, but other stem cells as well. It also kills the bulk tumor cells. So you get killing and antigen expression from the full set of cells within the tumor. Now that led to the trial of G47 delta by Tomoki Todo, former postdoc from our lab. He's now a professor at the University of Tokyo. It was a phase 1/2 trial, stereotactic injection, initially and then every 4 weeks, maximum 6 times, 3 times 10 to the 8th, or 1 times 10 to the 9th dose. And here, for example, is one of their better patients. Here's the glioblastoma, and you can see it over time until you get a complete response. And the 1-year survival was 84% versus 15% in the historical control. And this was designated a breakthrough drug in Japan, given orphan drug status, and then Daiichi Sankyo applied for the drug application, and it's been approved. It's now called Delicat, but it's basically been approved just like our FDA would approve it in the United States, but this would be FDA equivalent in Japan. So why does this work? Well, tumors that progress have evolved to evade the immune detection system. Oncolytic viruses can overcome them. Tumors die by non-inflammatory, non-antibody apoptosis generally. Oncolytic viruses can induce inflammatory cell death. Tumors have a defective danger signal PAM stains. Oncolytic viruses can induce pro-inflammatory DAMP symptoms. Tumors can induce immune tolerance. Oncolytic viruses can break tolerance, elicit innate and adaptive immune responses. Tumor-associated macrophages can be programmed to promote suppressor T cells. Oncolytic viruses can reprogram the tumor microenvironment, including tumor-associated macrophages. So for a number of reasons, this would appear to work. When we began studying this, we used a mouse 005 glioblastoma stem cell model. This was developed by Muramoto and Inder Verma, and basically with the gene associated here. And basically, you get a tumor, you can get glioblastoma stem cells, you can transmit them in C57 black foot mice. And they self-renew. They have all the hallmarks of GSCs. They express stem cell markers, such as CD133. They're highly tumorigenic. They're highly invasive. And they're fairly fully immunogenic. Very low MHC1 expression. We can reduce interferon. Absent MHC2 expression. PD1 is relatively low, but can be induced further with interferon. Now, oncolytic viruses are ideal for delivery of anti-cancer immune modulators that are toxic if given systemically. So a good one to look at is IL-12, which I've already mentioned. IL-12 is anti-angiogenic, pro-inflammatory, it promotes proliferation of T and NK cells, stimulates Th1 differentiation, induces interferon gamma production. It's been tried clinically, but it's toxic when given systemically. So IL-12 was sort of abandoned as something that could be used, even though it's very, very effective in terms of an immune modulator. So Tuba Chima was a postdoc in the lab, and she took G47 delta and made an IL-12 expressing vector, the CMV promoter, mouse IL-12 in this case. We now have human IL-12 as well. And then she made an empty virus cell with no IL-12 in it. And basically, IL-12 expression in these old flat tumors normalizes the vasculature, decreases Treg, and increases survival. So here's the vasculature, and you can actually visually see the difference between PBS, the empty virus, and the IL-12 virus. It's quantitative here. And if you look at Treg expression, PBS, empty virus, IL-12 virus, a significant reduction. And if you look at percent survival, with IL-12 compared to anti-virus, you get an extension of survival. And we could show that the anti-tumor activity was, again, dependent on T cells. The next thing to come along, as you've already heard from Dr. Sauer and others, is this idea of immune checkpoint blockade. And the first ones to be explored, as have already been discussed, were PD-1, PD-L1 system, and CTLA-4. And antibodies have been developed by various companies to block this. So glioblastoma, as you heard from Dr. Chan yesterday, remember one graph he showed, there were all these responsive tumors, and then glioblastoma was really an outlier. And in various studies using checkpoint inhibitors, glioblastoma has not been responsive. And there's been this concept of so-called immunologically hot tumors that have a fairly high mutational load of neoantigens. And then cold tumor, the glioblastoma is somewhere in between, but basically, you might as well consider it cold tumor because immune checkpoint inhibition does not work. And glioblastoma has low PD-1 expression, dominance of myeloid suppressive cells, multiple suppressive molecules secreted by the leukemic cells. Adenosine, for example, is 100 or up to 1,000 times greater in the tumor compared to normal brain and can immune suppressor. And there's T cell sequestration in bone marrow. So we began looking at this, and we basically began looking at combination of an IL-12 producing virus, G47 delta WBD, in combination with either anti-PD-1 or anti-CTLA-4. And basically what was done here was one injection of virus and then 3 injections with the antibody. And you see an extension both with the purple line, which is the virus, and with the anti-PD-1, extension of survival. And you see a slightly more extension if you put the two together, but still they all die. CTLA-4 is a little better. You get an extension with the virus, you get an extension with CTLA-4. You put them together, and you get one long-term survivor, but basically it's still not very good. So we hypothesized that blocking 2 independent immune inhibitory pathways might enhance the efficiency. And that was done, and basically, again, 1 viral injection, 3 antibody injections. So if you put anti-PD-1 and anti-CTLA-4 together, you get an extension of survival, but they still all die. If you add the virus in with them, you get 90% long-term survivors, complete remission. So that looks very positive. The Franco Saha was the postdoc in with them. And that was very prominent. And this can be repeated with this is the old 5 GSC model, re-challenging by injecting in the other hemisphere, show that you get no tumor. So there's a memory effect. And it can be repeated with another tumor called CT2A. where you get a long-term survival, and you use this so-called triple therapy of virus plus anti-PD-1, anti-CTLA-4, and again, immune memory with no tumor coming to be challenging. Now, it's not just the lymphocyte that are important. I'll show you a graph showing that. But we found that macrophage polarization contributes to eradication. And with this therapy, you get a conversion from MTA1, so you get a conversion from pro-tumor macrophages to anti-tumor macrophages. And that's now been shown by another group using a different virus, the herpes virus called 716, and not using brain tumors, but using mammary tumors. So macrophages become important. And we could show, for example, that if you treated either CD4, CD8, or macrophages, you could diminish the therapeutic effect. So this is with everything intact. If you deplete CD8 cells, which is the blue line, you get a diminution. If you deplete CD4 cells, you get a diminution. And if you deplete macrophages, you get a diminution. So it's a complex immune response. Although what I'll show you here, because it relates to another slide I'll show you, the most profound decrement was with taking away CD4 cells, which kind of surprised us. We thought CD8 cells would be more involved. But it turns out CD4 may be more important. And there was this recent paper from the University of Alabama group just in January, 2 months ago, showing oncolytic reprogramming of tumor microenvironment shapes CD4 T-cell memory via the IL-6 RA BCL16 axis for targeted control of glioblastoma. And you can show here that they use this virus, which is an IL-12 expressing virus, expressing amino IL-12 called MOL2. And basically, you have an extension of the virus, and if you get rid of CD4 cells, you get rid of it. And they could show that in human tumors, that if you had CD4 MHC2 high versus low, there was a difference in survival. So that basically, and I'll refer you to the paper, very complex paper. I won't go through everything in it, but basically, the oncolytic virus can reprogram the system in terms of increasing memory cells, increasing effector cells, interacting with glia, and increasing tumor killing. So basically, what we're talking about is taking a relatively cold immunologic tumor and using the virus to convert it to a hot tumor so that it would respond now to CTLA-4 or PD-1 or potentially other immune checkpoint blockers. Now, this works in other cancers as well, and I'll just mention two papers that were the initial ones done with TVEC, which is a virus expressing GM-CSF based on the studies that I've shown you, plus immune checkpoint inhibition therapy in metastatic melanoma. This one by Rebus, virus TVEC plus immune checkpoint inhibitor, anti-PD-1 in this case, double the response rate relative to anti-PD-1 therapy alone. And another by Chesney in Immunology, virus plus checkpoint inhibitor double the objective response rate compared to anti-CTLA-4 alone, 39 versus 18%, and diminished non-injected visceral lesions, 52 versus 23%. So this works for a variety of tumors in animal models and now in humans. This is a rich platform for development. Here's a partial list of other oncolytic viruses in clinical trial. I won't read them out, you can see them here. And here's a partial list of other immune checkpoint inhibitors and modulators. PD-1 and CTLA-4 were some of the first ones, but there are a whole host of others, and there are a whole host of other cytokines that can be looked at. And it overcomes this toxicity. If you start putting in these anti-PD-1, anti-CTLA-4, you get various immune effects with myocarditis, pneumonitis, colitis. By expressing some of them in the tumor, you may be able to avoid this. So what about other strategies? One would be to have the virus express the checkpoint inhibitors and load it. I'll show you an example of that. Another would be to consider intravenous therapy, immunotherapy. And the other would be synergies with other therapies. Now, for lack of time, I won't go into chemotherapy, radiation therapy. There are any number of papers showing synergy with various chemotherapies. I will mention something about synergies with other immunotherapy. Well, this has not been published yet, but that was presented at ASCO last year from the REPLA meeting. It's an open label study of a virus called RP2 alone or combined with nivolumab in patients with melanoma. And the data suggests that RP2, which expresses anti-CTLA-4 antibody, so this is again an example of now putting the immune checkpoint blocker in the virus. Those alone in combination have favorable safety profile with durable anti-tumor response. The basic idea here is you put the CTLA-4 in the virus, you give the PD-1 systemically, it avoids the toxicity of having both of them systemically. There was an objective response rate of 30% and disease control rate of almost 60%. And they can show the immune cell infiltration, et cetera. I'm sure this will be published in the near future. What about intravenous administration? Probably the most profound example of this was shown by Steve Russell and his colleagues using a measles virus expressing sodium iodide is important for labeling purposes. And basically, here's a patient with plasmacytoma. You can see it in the tumor up in the skull up here. And after viral infusion intravenously, you basically see it gone. This patient had a long-term remission. It's also been shown more recently in 2005 by Zhang et al. Using a recombinant Newcastle disease virus. So IV therapy is really what many people, including drug companies, would like to see. The problems are you get dilution of the virus, so the amount that goes to the tumor is far less than if you inject the tumor directly. You get non-specific absorption by blood vessels, by liver, etc. You get innate and acquired immunity, which you get rid of it in the bloodstream if you have antibody. And then there's the whole issue of tissue specificity. So it's been a significant problem, and most studies have been with direct injection. And I just might add, with current radiologic techniques, CT scan and MRI scan, you can inject a tumor anywhere in the body, pretty straightforward. So the other thing was combination therapies. And I'm not going to mention chemo or radiotherapy, but there are many studies in animals and in humans on that. But CAR T cells and mRNA vaccines are recently looking at, people have been looking at. And here's an article in 2022, Oncolytic virus-mediated expansion of CAR T cells improves efficacy. This is in animals now. And they use vesicular stomatitis virus, VSV, or real virus, and have prolonged survival of mice with subcutaneous melanoma and with intracranial gliomas. So the concept of putting oncolytic viruses in conjunction with CAR T cells is being explored by multiple people. There's a paper showing that IL-12 can potentiate CAR T cells, so obviously IL-12 can be used in the virus. And there are other ways of engineering the virus that can work with CAR T cells. There's another paper by Fu in signal transduction targeted therapy from last year. Combination therapy with oncolytic virus and T cells or mRNA vaccine amplifies anti-tumor effects. And this is in a B16 melanoma tumor-bearing mice. Combination therapy showed superior anti-tumor effects to monotherapy. Combination of oncolytic virus and mRNA vaccine displayed significant reduction in tumor burden and prolonged survival. So these concepts are being looked at by a number of different investigators. The other thing is, if you think about chemotherapy, you rarely give one chemotherapy section. Combination chemotherapy is standard for most tumor cells and most tumor types. So the idea of looking at more than one virus is something that is also being explored. Here's a paper in Nature Communications from last year. They engineered a rabbit virus, BSV, to express a truncated targeted antigen, which allows for HER2 targeting with this drug. And then they combined this BSV HER2 with an oncolytic vaccinia virus expressing HER2 targeted T cell engagement. The dual virus therapeutic strategy demonstrates potent curative efficacy in vivo in mice using CD3 to infiltrate their anti-tumor immunity. So basically, one can start looking at combining this, and there are a number of labs that are looking at this possibility. And you can imagine you can have a virus that attacks the tumor cell, a virus that changes the microenvironment. There are various ways of doing it. So I'm going to, as I end, mention some what I think are notable novel studies that are not yet published. There was an Oncolytic virus meeting in Rotterdam last year. It's held every year. And they just published the synopsis of this in Immunotherapy Oncology just a few weeks ago. And I just mentioned a couple of papers. There are many of them. But Altamonte from Munich is studying chimeric oncolytic virus using vesicular stomatitis virus and Newcastle disease virus expressing soluble PD-1. Again, two viruses. Steve Russell from the very end of the clinic showed a CMV engineering as a delivery vehicle capable of generating CAR T cells directly within tumor. Sadler mentioned this the other day. And so the idea here would be, instead of having to engineer CAR T cells in the patient, you have an off-the-shelf drug, in this case a virus, that would make CAR T cells directly within the patient. And you can use it for any patient. You don't have to change it. You don't have to do it specifically for one person. Here's another one from Barone from Candel Therapeutics, using an adenovirus, increasing overall median survival in patients with non-small cell lung cancer refractory PD-1. And then a very interesting paper by Dane in the presentation, they're not papers yet, using MQ710, which is the modified vaccine virus, and using multiple doses combined with pembro, using complete clinical and tumor pathological responses for precocious sarcoma. These are all exciting things. They're not yet published, but I'm sure you'll see them within the next year. And then finally, I'll end with one study that I thought was very exciting. This was not at the oncolytic virus meeting, but rather at the urology meeting. Phase 3 study of intravesical intrabladder virus. These names are so complex that I call this cretostimogene yada yada yada, for the treatment of high-risk BCG unresponsive. BCG is the standard treatment of non-muscle invading bladder cancer. So this is an adenovirus expressing GM-CSF. It binds the Coxsackie adeno receptor, which is expressed in all stages of bladder cancer. So it's a phase 3 study. There's an induction, secondary induction for non-responders, and then a maintenance. And this is given directly with catheters into the bladder. It's done as an outpatient, basically. They've had a 75% complete response rate. That is remarkable in that these are tumors that have not been responsive to the standard of therapy. And they're falling at 12 months and 24 months. And they get basically about half the patients that respond at 12 months. They're estimating there'll be about 40%, but they don't have all the patients followed up for 24 months. And at that point, I'm sure they'll publish it. But you can imagine, even if there are a certain number of recurrences, colleagues of mine, for example, G47 Delta is being explored in bladder and prostate cancer in Japan by someone who's a postdoc at Hiro Fukihara in our lab previously, he's a neurologist in Japan. And other viruses are being looked at as well in terms of bladder cancer. And so you can imagine treating one virus, you can get a recurrence, you can use another virus, et cetera. And I think that'll be one of the strategies being used. Now, one of the issues that have come up in multiple talks is public versus private investment. And multiple biotech companies now are getting involved, and big pharma is getting involved. Amgen announced that they took on this first viral therapy in U.S. TVEC. Bristol Myers Squibb, who I told you we're not interested when we first started this in 91, now have an exclusive worldwide license with Cyoxis for an oncolytic virus to address solid tumors. Merck is involved with a big oncolytic virus, since Amgen. Takeda teamed up with Termstone in a large deal to work on biotech. Novartis is collaborating with Gilead developing in vivo CAR T-cell therapies. So the big pharma is now putting money into this. And in fact, in looking at this for this talk, 3 viruses are currently in clinical trial. There's an adenovirus H1N1 in China, herpes 247 Delta in Japan, herpes virus TVEC in the USA. These are now being approved by the FDA in the United States or the FDA equivalent in these other countries. And there are now multiple ongoing oncolytic virus clinical trials. I looked in clinicaltrials.gov, a couple of weeks ago when I started doing this talk, and there are 166 studies ongoing that are mentioned in clinical trials. Hanson Wade follows the business aspect of this, and they mentioned in the pipeline there are 701 tracked assets, 607 are currently active, 5% are in late-stage clinical development, so many of these are early-stage trials. And there are many investments in deals being made, strategic partnerships obviously continue to be critical. So what I've tried to do in this talk is show you that this platform of oncolytic viruses is very rich. It's iterative, multifaceted, it's synergistic. The iterative in math and mean applies to applying the function repeatedly using the output of the iteration, the input of the next. Here, it's bench to bedside, back to bench, back to bedside. So you continue to improve on it, as I've shown you. It's multifaceted. We initially started this looking at oncolytic. We've shown that it's not affected by hypoxia. It can be tumor cell specific, because it's specific to motor vein. It can kill tumor stem cells. It can be anti-angiogenic. And as I've shown you, it can be very immunogenic. And it's synergistic with chemotherapy, radiotherapy, anti-angiogenic drugs, immune checkpoint blockade, cytokines, and as I've shown you, CAR T cells. So it's a very, very rich system that is still being developed. In ending, I'll just mention thanks to two virologists I've worked with on, one who worked with DLSBTK and Sam Rampton, who's worked at the SIN. There are a number of postdocs involved. Yoko worked in the initial stuff. He's now chairman and professor at Harvard and master of Brigham. Jim Marker worked on the G207 work and is now chairman and professor at the University of Alabama. Wilkie Toto was a postdoc. I told you he did the stuff in G47 development. He's now professor at the University of Tokyo. And Masahiro Toto did the stuff with the initial stuff with viral immunotherapy and cytokines. And he's now chairman and professor at Keio University in Tokyo. And with that, there have been a number of other people. I won't mention them in detail, but they're all very thankful to all of them. With that, I'll end and leave it open to questions.